Hello, and thank you for joining us for Chardan's Virtual Ophthalmology Summit, where the focus in this installment will be on rare ophthalmic diseases. My name is Daniil Gataulin. I'm one of the senior biotech analysts here at Chardan, and it is my pleasure to introduce Natalia Misciattelli, CEO of AAVantgarde Bio. Natalia, thank you for joining us today. The
Having
session
me.
will follow. Yeah, thank you. The session will follow a fireside chat format. So to start things off, can you spend a few minutes to introduce yourself and AAVantgarde Bio to the audience?
Yes, so Natalia Misciattelli, I'm the CEO of AAVantgarde. We are a gene therapy company focused on large gene delivery with two very exciting programs in IRDs that will shortly both be in the clinic.
Excellent. And then just to give a little bit of a background about your company, when was it formed, where you're located, and to date, who have been the main investors and the funding that you've been able to raise to date?
Very good question. So we are Italian of Italian origin, with an international team now. We have a team in Italy, a large team in the UK and in the US. We were formed in 2021, and in terms of backers, we're backed by Sofinnova Teleton, Atlas, Forbion, and Longwood in our Series A. We raised $65 million in our Series A.
Got it. Excellent. So let's talk a little bit about your program. So you're focusing on developing two platforms to address the challenge of delivering larger genes using AAV gene therapy. So your first platform is a dual hybrid AAV platform. Your second platform is INTIN-mediated protein transsplicing. So let's talk about the dual hybrid platform first. Can you explain what it is or rationale behind this approach?
Yeah, so the dual hybrid platform was really developed to try and overcome the carrying capacity issue in AAV gene therapy. And the platform's aim is to overall, our platform is delivery of oversized genes, and we really have two different technologies that lies below that. So one is the dual hybrid, and the other one is the INTIN mediated. And for us, our focus is as high as possible expression to ensure that we get the best possible impact for the patients in the clinic. So depending on what gene we're looking to express, we will pick the best platform technology for that particular gene.
Got it. Okay. So obviously it's a challenge, right, to get larger genes and that into the cells, and that approach allows you to overcome this. Can you talk about some of the specific key technical challenges that you had to overcome to maximize the efficacy as well as safety of this approach?
Sure. So the concept or the idea of DNA splicing is quite intuitive. If you'd want to express a larger gene, it doesn't fit in an AAV capsid, you might try and intuitively think, well, maybe if I split it in two, I could splice it together. The challenge is to do that at a high enough efficiency so that you don't end up having to push to a very, very high dose, because you get a small proportion of genes that are spliced together. So that was really the driver of finding the right way of doing this, which is really down to the splitting points to ensure that you get high efficiency. So that's what our proprietary platform technology does, is pushing that efficiency level to ensure that we can take the programs into the clinic and that you don't end up in a situation where it's actually prohibitive, the dose, in terms of inflammation or other issues.
Got it. That makes sense. And in terms of as you continue to work and develop this, what do you think continue to be the main limitations of this approach?
It's a good question. Limitations, we haven't to date really found many limitations, but what we are always trying to do is we're always trying to optimize, as I mentioned, for efficiency. So for the dual hybrid, there will be situations where the dual hybrid technology is not allowing us to be expressing genes at a high enough proportion, and then we may change to our second technology, which is our INTEEN mediated platform, and that's indeed what we've done with the Stargardt's program. So we're using for Stargardt's, we're using INTEEN technology, and for our Usher's program, Usher 1B, we're using our dual hybrid. So it really is a question of what gene, and ensuring that we get the best possible results ultimately for the patients.
Okay, got it. Yeah, that makes sense. And but we'll of course spend some time to talk about your other approach in a few minutes too. But back to this one. So with multiple vectors, generally transduction efficiency seems to decrease. And then expression of a full-length product after it's been spliced also depends on recombination efficiency once separate vectors are inside the cells. So how do you maximize, without disclosing your proprietary science, but how do you maximize transduction and recombination efficiency, and still keeping the dose down to not compromise safety?
Yeah, it's really down to the splitting points. And there is research out there that's actually compared our splitting points with other available innovation. And we're pleased to see that we always come out on top. And that's probably the reason why there are also actually a couple of other programs out using our splitting technology in clinical trials at the moment. There's 3 other clinical trials using our technology that's all recruiting patients as we speak. One is the Ottof program for Akouos and stroke Lilly.
Got it. Okay. All right. So you mentioned that your dual hybrid approach, you're utilizing it for Usher 1B. So let's talk about that indication. So can you talk about what Usher 1B is? How this disease is manifested? What is the disease prevalence in US and Europe? Any helpful background on the indication, please.
Yeah. So Usher 1B is an inherited retinal disease. It's a disease that manifests with a double disability, both a hearing loss and a vision loss. The hearing loss comes first. So patients are normally identified because of the hearing loss that tends to already be pronounced at birth. So the patients are therefore identified because of the hearing loss. And once they're genotyped and it's discovered it's USH1B, of course, the prognosis will be that their sight will also gradually be lost. In terms of the disease itself, it's one of the larger IRDs. There are really 4 large IRDs that are out there. Stargardt and USH1B is 2 of the large 4.
Okay, got it. And with this approach, with your approach, what fraction of Usher 1B patients are potentially addressable?
Yeah, it's an excellent question. So you could be tempted to also look at the hearing loss and the eye. For the hearing element, because that happens very early, our assessment is that if we can save the sight for this patient population, that will be meaningful. So we're focused on the eye indication for USH1B, and really based on our natural history study that we've conducted, we know that patients still have photoreceptors remaining, not a lot of sight, but still some photoreceptors remaining quite late in life. So it will be quite possible to treat both early and late because of the patient's sort of decline over a lifetime. And it's similar to what we've seen with Luxturna, that even though that's a disease of adolescence, actually photoreceptors do remain till quite late, that give the possibility of treating patients actually quite late in life.
Okay, got it. Understand. And you referred to the natural history study. I understand it's called Stella study. So if you can highlight some of the key learnings. You talked about the disease progression and the fact that some people have receptors that are still functioning in at least early adulthood. Um, can you talk about other key learnings that helped you design your clinical program and maybe some of the other um, important points about that disease and the disease progression?
Sure. So our natural history study for ASH1B was conducted and completed. Our natural history study for Stella is actually for our Stargardt's program. But in terms of learning from our first natural history study, what we learned, first of all, was that this patient population is a patient population because of their double disability. We needed to make sure that we considered the patient's ophthalmic examinations. In part, sometimes are using hearing, beeps, guidance, etc., etc. So actually, that was one of the big learnings, was making sure that once we got into a clinical study, that actually we had considered the limitations due to the hearing issues. The second part we learnt was really understanding the patient population, understanding their progression, understanding each individual patient, understanding which ones were more suited for an interventional study, and also engaging the community through the natural history study. So we're firm believers in a natural history study provides you a lot of insights, but it also is a great opportunity to engage with the community. And we've published the data as well to share that data set.
Got it. Okay, excellent. Yeah, makes sense. So the clinical study. So I believe that's ongoing. Can you talk about the trial design and maybe to kind of give us an idea of the status? When was the first patient dosed, and how many total patients and at what doses have been treated to date?
Yeah, so we announced our first patient dosed in September last year, and the last public update we gave, we had dosed 8 patients, and that is a 15-patient study. It's a standard dose escalation, 3 different doses, and we are fully expecting to have enrolled all our patients by the end of this year. So we're progressing very rapidly. The data set is maturing. We are ensuring that we're sharing that data set with the community, and we will be presenting data at all the expected upcoming ophthalmic conferences to make sure that the community gets to hear about the progress that we've been making. The last update we made, which was a safety update from our first 8 patients, we showed a lot of safety data because, of course, what's worth stopping and considering is that we are the first company to take a dual AAV into the clinic, the first company to show that transduction is possible, and that we're not seeing anything unusual from a safety point of view. So we're very happy with the safety data that we presented. We shared a little bit of data from our first patient. Of course, it's early days, but we will continue to show that data set as the data set matures. But we're very excited about the data generated to date.
Got it. Okay. And can you tell us about the patient population, how old, and their baseline, and what endpoints, efficacy endpoints are you looking at?
Yeah, so of the patients we've treated to date, they're all adults, and we have treated patients in the first 3 decades of adult life. It's a variety, male, female, And what we've also done is we've also ensured that we have looked at patients with a sort of a breadth of disease manifestation. So we will gather a really meaningful data set.
Okay, got it. You mentioned the last update was, I believe, at ARVO, right, in May.
Correct. Yeah.
And so as you mentioned, safety looks clean. And in terms of efficacy endpoints, can you talk about some particular observations that you're most excited about?
Yeah. So I mean, for our phase 1/2, obviously our primary endpoint is safety. However, we are also considering a multitude of secondary endpoints to look for potential efficacy signal. We're looking at all the standards that you would expect. So, we're doing BCVA, LLVA, microperimetry. We're doing virtual maze. We're doing the standards. What I would say that we're probably of the data set we presented at ARVO, we shared data on BCVA, LLVA, and microperimetry. These were the three that was really made public, and that really is a question of, as the data set matures, those were the first ones that made sense to share, but there will be more to come.
Okay, makes sense. Who would you say are the main competitors for Usher 1B program currently? And if there are any data from those programs, what are some of the key learnings that you're taking away from those data?
So there's no one else who's in the clinic, so that, and I guess in terms of learnings, there isn't really a lot of learnings from others. But there's a lot of responsibility to make sure that that we progress this program and that we don't let down this patient population who are very underserved. What we're finding is that because of the double disability, it is of course highly detrimental to lose your sight if you already have issues with your hearing and actually also your vestibular balance. So the patients are very forthcoming, very enthusiastic to work and understand and to help their own community. So there's more of a responsibility rather than a learning at this point.
Got it. Okay. And to round up this discussion about Usher 1B, what are the next steps and what potential milestones in the next 6, 12, 18 months?
Yeah, so we will, as mentioned, we will have enrolled the full study by the end of this year, and we will continue to publish at all the big ophthalmology conferences, and we'll be sharing the data set. So that's really where you would expect us to be. As the data set matures, we will be sharing it. And that's really our upcoming milestones with Ushers. It's a program that's progressing very fast. As you can hear, we have, by May we had 8 patients. We started in September, so we're progressing very rapidly.
Got it. Excellent. All right, so let's switch gears and talk about your second program, StarGarde, that relies on the INTENE platform. So first, similar to my previous question, can you walk us through this Stargardt disease pathology for the patients when they get diagnosed and how they manifest?
Yeah, so Stargardt's patients are often diagnosed in childhood. It's a progressive loss of vision, and the patients. It's the most common inherited retinal disease, so there's quite a lot of Stargardt's patients out there, and from a community point of view, the Stargardt's community have not had any treatment. There's been no treatment at all available to them. There's no approved treatment, and yeah, so I think it's a disease that's highly underserved.
Hmm. Got it. Makes sense. And so the approach that you're using for this indication is INTIN mediated. Can you talk about what that means, science behind it, and why that approach works better or is more appropriate for Stargardt than the other?
Yeah, so it really it comes back to the idea of you need to find the large gene technology that works the best. And not all genes are the same for obvious reasons. And so therefore you need to identify what technology should you be using and for what. The INTIN technology is incredibly efficient. So we use that for the Stargardt's program. We've quantified the efficiency of that platform. It's 92% as efficient as a single AAV, so it's incredibly efficient. And actually, we are very proud to be the forefathers of this concept in gene therapy. We have also published extensively. We have done a lot of work on INTIN technology. We've just recently published a beautiful publication in Science Advances that shows our large animal data, both in NHPs and in knockout pigs, looking at quantification of the platform. So really looking at one thing is what happens to mice, but what happens in a relevant animal model? What happens in a knockout pig where you can really look at the expression level and it will translate into the clinic. So we're really pleased with that publication. We're also really pleased with our we just recently completed also some more data to be able to compare between the efficiency in general. And what we're seeing is that the platform, the INTIN platform for Stargardt's is really working out very, very nicely. So we're expecting to get into the clinic at relatively low doses because of the efficiency of the platform. So I think that's something to look out for as we get into the clinic is really that ability to have a platform that will be truly efficient and really be able to make a big difference in the clinic. So we're excited to be getting into the clinic this year.
Mm-hmm. Yeah, excellent. And so certainly sounds like there is a lot of promise, a lot of potential with this approach. Can you share some potential limitations in what stations it would not be suitable or not quite as efficient?
Yeah, it's a great question because I think there are of course different what we've seen. There's not all patients are the same. There are certain opportunities depending on what platform technology you use. We're going back to the root cause of the disease. We're augmenting the gene. So therefore we can treat all patients with Stargardt's disease. The only limitation we really have is of course, we can't bring photoreceptors back that have already died. But what we can do is we can treat patients early and late, and we can treat all Stargardt's patients.
Makes sense. Okay. And to close the discussion on Stargardt, you mentioned that your goal is to be in the clinic later this year. Can you give us maybe a bit more granular timeline of the next steps and what to expect for that program?
Yeah, so I think what I can say is there will be news coming, and we're a company that has experience of running IRD trials. I think having one program in the clinic already and recruiting well, we're certainly intending to run our program as efficiently as our first program What I can tell you is that our natural history study called Stella, we have already enrolled our part A of that. So we have all the patients from our natural history study, which is, of course, a huge advantage as we go into the clinic. And we're expecting that we'll be in the clinic in 25, as I mentioned.
Got it. Excellent. All right. Let's shift gears and talk more about some of the general items that pertain to both of your programs. So first, can you tell us a little bit about your CMC strategy as you continue to advance your SURE-1B program and as you're getting ready to advance Stargardt into the clinic?
Yeah, so for us, our CMC strategy, we're working with CDMOs. Most people are. We're not investing in an in-house manufacturing for the simple reason that that the doses that we will need will not warrant in-house manufacturing. So that's our strategy, is to outsource. We have outsourced to expert CDMOs. We're working very closely with them in partnership, and we'll continue to do so.
Got it. Okay. And the product that's manufactured for those clinical trials, would that be the same product and the same CDMO that would eventually manufacture the product for pivotal and commercial?
I think that it remains to be seen. I think the most important thing is to manufacture high quality products. It's also important to consider that these products are dual vector, but they should be one IMP. We need to make sure that we've thought through how the CMC, how the product is going to be utilized in a hospital setting, and so therefore we're working closely with our CDMOs to make sure that they can deliver that one IMP.
Okay, makes sense. Also, as you mentioned, there aren't any approved products for either of those indications. The regulatory landscape is quite novel, right? There is no precedent, and there potentially seems like there's an opportunity for some flexibility or for some novel approaches in terms of endpoints, in terms of other items that you can use, develop, and implement to maximize the chances of success for your programs. Can you talk about some of the things that you're looking into, that you're considering? Maybe virtual maze, maybe goggles for home use, or anything else that is new and exciting?
Yeah, so I fully agree with you. I think gene therapy is an exciting field, and I think the opportunity to really treat a disease going very much back to the basics of treating the root cause of the disease is exciting, and it's exciting to regulators too. So we've been having conversations with regulators both in the US and in Europe and in the UK to make sure that we really understand what the opportunities are, that we understand the openness for certain new approaches, and we found the regulators very receptive. And I think we've just recently seen in the FDA the listening tour, and we think that this is a really fantastic opportunity to push some of the novel approaches, as you mentioned. So I think there's more to come, and I think as a community, so gene therapy community and an ophthalmology community, will probably make sure that, well, I hope that we'll work together to make sure that we all progress.
Okay, great. And speaking of regulators, given the changes at the FDA in the past couple of months, are you seeing any changes in your interactions with US regulators?
We haven't seen anything. We haven't seen anything other than support and positivity. So yeah, we're very happy.
Excellent. A couple more questions to finish off this discussion. Um, can you talk a little bit about the core team at AAVantgarde who are helping to bring the company's vision forward?
I'm so pleased you asked about that because I think many times we spend a lot of time talking just about the science, but the reality is that it's the team that brings the science alive. Um, and and so you need a team that is expert-led, that understands what they're doing, and are passionate about what we do. So little by little, we've been building a team. Our scientific founder, Alberto Riccio, is still very much involved with us. And then we've been building an expert team. We recently hired a CMO who has deep expertise in IRDs, understands endpoints very well, has been working on endpoint development, also looking at novel endpoints and really understanding those disease areas in depth. CMC is the same. You really need experts CMC, and again we have hired a fantastic hire there who is helping us stay on course, understanding what's important, helping us move not just at pace but also high quality and a thoughtful CMC strategy. And then in addition to that, we've got a full corporate team who's also been supporting us and driving the company forward along with regulatory. So we've got a full team now on board that's very expert. And I'm very confident that this expertise will not just drive us forward, but also drive us forward with innovation.
Excellent. Great. That's great to hear. And so one last question that we like to end our discussions with. What do you think investors are still missing about the AAVantgarde story?
That is a great question. I think I think investors are excited to see what's to come. I think we're all excited to see what's to come. In the end, we all want to help patients have meaningful treatments. And I'm a firm believer in if the treatment is meaningful, then everything else falls into place. So as we publish our data for USH1B, and as we begin to generate the data for Stargardt's, I think there's a lot to come. And I think there's a lot of people, not just patients, but there's a big community watching. So we're looking forward to sharing.
Excellent. Great. Well, with that, I think we are at the end of our session. Again, Natalia, I appreciate you joining us. It was great chatting with you. Thank you.
Thank you so much.